Welcome, deep divers. Today we're taking you on, well, an incredible journey, right into the most fundamental unit of life, the cell.
It's fundamental, but also profoundly complex.
Exactly. Imagine trying to understand the blueprint for, like, all living things. That's basically our mission today. We're cracking open a chapter from Marks Basic Medical Biochemistry.
A classic text.
Yeah, and we want to distill the absolute essentials of cell biology and biochemistry for you. Our goal isn't just to tell you what the cell's made of, but really to uncover the sheer genius of its design.
And how its intricate machinery really forms the foundation of health and disease.
Right. Think of it as your essential guide to understanding this microscopic universe that makes you, well, you.
Precisely. And in this deep dive, we're going to explore the cell's elegant architecture, how it interacts quite skillfully with its environment, and the highly specialized roles of its internal components.
And you mentioned connecting these to clinical examples.
Absolutely. We'll connect these biochemical pathways directly to real-world clinical cases. That helps you see how these, sometimes abstract cellular processes actually play out in human health.
Making it tangible.
Exactly. Making these core concepts not just understandable, but hopefully truly unforgettable, helping students grasp those crucial links between how cells work and medical outcomes.
Okay, let's do it. So let's zoom in on the cell, a universe in miniature. We hear it called the basic unit of life all the time.
Right.
But what does that really mean, especially when you think about, I don't know, the sheer diversity, nerve cells, skin cells, they're so different.
That's a great question. Because while human cells are incredibly diverse, each one adapted for its job, they all share this remarkable set of common architectural features.
Okay.
We classify human cells as eukaryotes. That means they all have a defining outer boundary, the plasma membrane.
Right.
And inside that, they have internal membranes that enclose the nucleus and other specialized compartments, the organelles.
Organelles, got it.
And holding it all together, sort of structuring it, is this dynamic internal scaffolding, the cytoskeleton.
Okay.
Now, this eukaryotic structure with its membrane-enclosed nucleus holding the DNA, that's a really crucial distinction from prokaryotes.
Like bacteria.
Exactly, like bacteria, they lack these internal compartments.
Yeah.
And understanding this difference is just vital in medicine. Think about antibiotics. Many target those bacterial unique features.
That makes total sense. Okay, so if the cell's a mini-universe, that outermost boundary, the plasma membrane, you said, it's not just a simple wall, it's more like a gatekeeper.
An incredibly sophisticated gatekeeper, yes. The plasma membrane is this lipid bilayer, often described using the fluid mosaic model.
Fluid mosaic.
Okay. Imagine two layers of tiny molecules called phospholipids. Each one has a head part that loves water hydrophilic and a tail part that fears water hydrophobic fatty acyl chains.
Okay, water-loving heads, water-fearing tails.
Right. And they naturally arrange themselves with the heads facing the watery bit outside and inside the cell, and the tails huddling together in the middle, forming this oily core.
Makes sense.
Key phospholipids here are things like phosphatidylcholine, phosphatidylethanolamine. Oh, and phosphatidylserine is interesting. It carries a net negative charge.
A negative charge.
Yeah, and it's mostly found on the inner leaflet, the side facing inside the cell. That asymmetry is really important for cell signaling.
Okay, so phospholipids make the basic barrier. What else is in there? You mentioned something playing a dual role earlier.
Ah, yes, cholesterol. It's slotted in between those phospholipids, and it's absolutely crucial for maintaining the membrane's fluidity.
Fluidity, so keeping it flexible.
Exactly. Think of it like the cell's internal thermostat for flexibility. Without cholesterol, those fatty acid tails, especially the ones with kinks, the cis unsaturated ones, they'd pack too tightly.
Making it rigid.
Right, making the membrane too rigid. Cholesterol prevents that, allowing lipids and proteins to move laterally within the membrane. And that movement is essential for things like cell division, cells squeezing through capillaries, forming vesicles.
So it's actually really important for the cell, not just something bad from our diet.
Absolutely vital for normal cell function, yes.
Okay, so a dynamic lipid foundation, cholesterol for flexibility. What about the proteins embedded in this mosaic?
Right. The proteins, they do a ton of critical jobs. You've got integral proteins. These guys span the entire membrane.
Go all the way through.
Yep, with their hydrophobic parts tucked into the lipid core and their hydrophilic ends sticking out into the water. They often act as channels or transporters or receptors.
Okay.
Then you have peripheral proteins. They're just attached to the surface, usually by weaker electrostatic bonds, maybe to the lipids or to the integral proteins.
Like spectrin.
Exactly. The spectrin family is a classic example, like dystrophin in muscle cells. Defects there cause muscular dystrophies, devastating conditions. And finally, there are lipid-anchored proteins. These are covalently attached to lipids on either the inner or outer surface. The prion protein, for example, uses a GPI anchor.
A GPI anchor.
Yeah, glycosylphosphatidylinositol. It's basically a specialized lipid tail that moors the protein to the outer membrane surface.
Okay, and what about that fuzzy coat you sometimes see on the outside? The glycocalyx.
Ah, the glycocalyx. That's a fascinating carbohydrate layer on the outer surface, made of glycoproteins and glycolipids.
What does it do?
Well, it acts as a protective shield, helps restrict uptake of hydrophobic stuff. But maybe even more importantly, those specific carbohydrate chains act as critical cell recognition markers.
Like ID tags.
Sort of, yeah. Think about blood groups A, B, O. Those are determined by the unique carbs on red blood cell surfaces. Pathogens, though, can sometimes hijack these for entry.
Speaking of pathogens, that makes me think. You mentioned how damaging it is when this membrane gets compromised.
Yeah.
Like with Clostridium perfringens, gas gangrene.
Yes, that's a truly catastrophic event for the cell. That bacterium produces a lipase, an enzyme, that specifically attacks and hydrolyzes key phospholipids, like phosphatidylcholine.
So it just chews up the membrane.
Effectively, yes,
Yeah.
tears holes in it. This leads to cell lysis. The cell just bursts open, spills its contents, and those contents then feed the bacteria, fueling the infection.
Horrifying. And you also mentioned alcohol affecting the membrane, like in LM's case.
Right, a different kind of impact. Ethanol molecules can actually intercalate, sort of wedge themselves between the membrane lipids. This subtly but significantly increases membrane fluidity.
Makes it more floppy.
In a way, yes. And that seemingly small change can profoundly affect the function of integral proteins embedded in the membrane, like ion channels and neurotransmitter receptors in the brain.
Which explains the immediate effects of intoxication.
Exactly. It alters nerve impulse conduction.
Wow, okay, so membrane structure is vital. But it's still a barrier, and cells need to get things in and out. That brings us to traffic control, transport across the plasma membrane. How do they manage this?
They have incredibly sophisticated transport systems. For small molecules, ions, we generally categorize them into four main types.
Four types, okay.
Simple diffusion, facilitative diffusion, gated channels, and active transport. And we can group these as passive, no energy needed, or active requires energy, usually ATP.
The bigger stuff.
For really large molecules or particles, they use a whole separate mechanism called vesicular transport.
Okay, let's start simple then. Simple diffusion.
Simplest of all, it's just movement directly across the lipid bilayer, purely down a concentration gradient. High concentration to low, no energy required.
What kind of things move like that?
Think gases, oxygen, carbon dioxide. Also lipid-soluble stuff like steroid hormones, they just slip right through. Water can too, sometimes directly, sometimes through specific protein pores called aquaporins.
Okay, that's for small, lipid-friendly things. What about molecules that are like too big or too charged to just slip through?
That's where facilitative diffusion comes in. Still passive, no direct energy needed, but it does require help from a specific transporter protein.
A helper protein.
Exactly. The molecule binds to this protein, the protein changes shape, and releases a molecule on the other side. It's neat because it shows saturation kinetics, like enzymes.
Meaning it can get overwhelmed.
Right. If all the transporters are busy, the rate maxes out. And it's highly specific. A glucose transporter moves glucose, not fructose, for example.
Okay. Now, you said gated channels.
Yeah.
This is where it got really interesting with that clinical case, Dennis the Thief and cholera, right? How do these gates work?
Gated channels are fascinating. They're transmembrane proteins forming a pore for ions, but they have an internal gate.
A literal gate.
Metaphorically, yes. It opens or closes in response to a specific trigger, a stimulus.
Like what?
Could be a change in voltage across the membrane. Those are voltage-gated channels, crucial for nerve impulses. Or binding of a specific molecule, ligand-gated channels. Or even phosphorylation adding a phosphate group.
Phosphorylation gated.
Yes, and the cystic fibrosis transmembrane conductance regulator, CFTR, is a perfect example of that. It's a phosphorylation-gated chloride channel.
And that's what goes wrong in cholera.
Precisely. In Dennis Fee's case, the cholera toxin binds to specific lipids, GM1 gangliosides, on his intestinal cell surface. It gets inside the cell, and ultimately it permanently activates CFTR by promoting its phosphorylation.
permanently activates it. What does that do?
It causes a massive uncontrolled flood of chloride ions out of the cell into the intestine. Sodium and water follow passively, leading to that severe watery diarrhea and dehydration.
Wow. And cystic fibrosis is the opposite problem.
Exactly. In CF, mutations mean the CFTR channel is inactive or missing, so chloride can't get out properly, leading to thick, dehydrated mucus in the lungs and elsewhere. Same channel, opposite problems.
Incredible how one channel is so critical. Okay, so those are passive mechanisms going down the concentration gradient. What if a cell needs to move something against the flow?
Now you need active transport, and this absolutely requires energy, usually from ATP hydrolysis.
Okay, got to spend some energy currency.
Right. Two main types. Primary active transport uses ATP directly to power the pump. The classic example is the Na plus, K plus, ATPs.
The sodium-potassium pump.
That's the one. Pumps 3 sodium ions out, 2 potassium ions in for every ATP used. Creates those vital ion gradients. Another key one is the Capro2 plus ATPase, pumping calcium out or into stores to keep intracellular levels very low.
Crucial for muscle contraction, right?
And signaling, yes. Then there's secondary active transport. This one's clever. It doesn't use ATP directly.
No. Then how does it work?
It harnesses the energy that's already stored in an existing ion gradient, often the sodium gradient set up by the primary pumps.
Okay, so it uses the gradient like a battery.
Essentially, yes.
Yep.
It uses the downhill movement of one ion, like sodium, to pull another compound uphill against its gradient.
Can you give an example?
The perfect clinical example is glucose transport into intestinal epithelial cells. Glucose gets co-transported with sodium. The sodium moving down its gradient provides the energy to pull glucose into the cell, even if glucose concentration is already higher inside.
Ah. And that's why oral rehydration therapy works for cholera, the glucose and sodium solution.
Precisely. The glucose helps drive the uptake of sodium, and water follows, rehydrating the patient. It leverages that exact secondary active transport mechanism.
Brilliant. Okay, and for really big things like bacteria or large proteins, you mentioned vesicular transport?
Yes, vesicular transport. This is where the cell membrane wraps around the substance to form a little bubble, a vesicle, which then pinches off.
Bringing stuff in is endocytosis.
Right, endocytosis. And there are different flavors. Phagocytosis for big particles like bacteria cell eating, pinocytosis for fluid cell drinking, receptor-mediated endocytosis for specific molecules that bind to receptors like LDL cholesterol uptake.
And podocytosis.
That's a special type using tiny indentations called caveolae, often for small molecules like folate. And the reverse process, moving stuff out in vesicles, is exocytosis.
Like releasing hormones or neurotransmitters.
Exactly. The vesicle fuses with the plasma membrane and releases its contents outside.
Okay, our journey continues deeper inside the cell now, beyond the membrane to the organelles, specialized compartments. These are the mini-cities within the cell, right? Each with its own job.
That's a great way to put it. Let's start with lysosomes, often called the cell's recycling centers, or maybe less appealingly, digestive factories.
Digestive factories.
Yeah, they're single membrane sacs filled with powerful hydrolytic enzymes, nucleuses for DNA, proteases like cathepsins for proteins, glycosidases, esterases, the works.
And they work in acid.
Exactly. These enzymes need a very acidic pH, around 5.5, to function. And lysosomes maintain that using special proton pumps, VATPases, that pump hydrogen ions in.
So do they digest?
Unwanted material, worn-out organelles, bacteria brought in by phagocytosis. They also recycle cellular components through a process called autophagy.
This reminds me of Lotta T's gout again. You said lysosomes were involved, but destructively.
Yes, it's a fascinating, albeit painful, example of things going wrong. Lotta T had high uric acid, forming sharp urate crystals in her joints. Immune cells, neutrophils, trying to clean them up by phagocytosis.
They eat the crystals.
They engulf them, yes. But the lysosomal enzymes can break down the crystals. Instead, the sharp crystals physically damage and rupture the lysosome membrane inside the neutrophil.
Oh, no.
Yeah. This releases all those powerful digestive enzymes right into the cell cytoplasm and eventually out into the joint space, triggering that intense inflammatory response of acute gout.
Ouch. And this relates to lysosomal storage diseases, too.
Directly. In those diseases, like Tay-Sachs or Pompe, there's a genetic defect in one specific lysosomal enzyme. So the material that enzyme is supposed to break down just accumulates and accumulates inside the lysosome, eventually wrecking the cell.
Sobering stuff. Okay, from waste disposal to the power plant, the mitochondria.
The famous powerhouses. They generate most of the cell's ATP, its energy currency, through fuel oxidation and oxidative phosphorylation.
They have two membranes, right?
They do. A highly impermeable inner membrane folded into these cristae. That's where the electron transport chain and ATP synthase, the real energy-making machinery, are located. Then there's an outer membrane with pores, porins, making it more permeable.
And inside.
Inside the inner membrane is the mitochondrial matrix. That's where enzymes for the TCA cycle, the Krebs cycle, hang out, central to fuel metabolism.
And they have their own DNA.
Uniquely, yes. A small amount of circular DNA inherited internally. This links them to various mitochondrial diseases and maybe even aging processes.
Interesting. What about the other oxidative organelles, the peroxisomes?
Peroxisomes. Often overlooked but vital. They're small organelles, single membrane, involved in oxidative reactions that produce hydrogen peroxide.
Which is toxic, isn't it?
Highly reactive, yes, but peroxisomes also contain enzymes like catalase that quickly break it down safely into water and oxygen.
What else do they do?
Really important things. Oxidizing very long-chain fatty acids, converting cholesterol to bile acids in the liver, synthesizing certain lipids called plasmalogens. Defects cause serious neurological diseases like adrenoleukodystrophy.
Okay. Now, the big boss,
Yeah.
the nucleus, the control center.
The largest organelle in animal cells, usually. It houses almost all the cell's genetic material, the DNA, which is organized into chromosomes with proteins called histones. That complex is chromatin.
And the nucleolus is inside there, too.
Yes, the nucleolus is a dense region within the nucleus. It's where ribosomal RNA, our RNA, is transcribed, and ribosomes are assembled before they get shipped out.
And the whole thing is wrapped in.
The nuclear envelope. It's actually a double membrane, 2 lipid bilayers separating the nucleus from the cytoplasm, and it's studded with nuclear pores.
Gates again.
Exactly. Selective gates that control traffic in and out. Messenger RNA and ribosomes need to get out to the cytoplasm for protein synthesis. Nuclear proteins, like DNA polymerase, need to get in, guided by specific tags, nuclear localization signals.
And extending out from that nuclear envelope is this big network, the endoplasmic reticulum, the ER.
That's right. The ER is this complex, interconnected network of membrane tubules and flattened sacs. Comes in 2 flavors.
Smooth and rough.
Exactly. The smooth ER, SER, looks smooth because it lacks ribosomes. It's a major site for lipid synthesis, making triacylglycerols, phospholipids, also crucial for making steroid hormones. And critically, it metabolizes many drugs and toxins using cytochrome P450 enzymes.
The P450s. And this ties back to LM and alcohol.
It's a direct link. Chronic alcohol use dramatically increases the amount of the microsomal ethanol oxidizing system, MEOS, in the liver SR. MEOS uses P450s to metabolize ethanol to acetaldehyde.
Which is toxic.
Very toxic. Acetaldehyde can inhibit other cellular processes like tubulin polymerization, which is needed to secrete fats from the liver in VLDL particles. This contributes to the fat buildup seen in fatty liver disease.
Okay, so that's the smooth ER. What about the rough ER?
The rough ER, RER, is rough because it's studded with ribosomes on its surface. This is where proteins destined for secretion or for insertion into membranes or for delivery to other organelles like lysosomes are synthesized.
They get made right there on the surface.
They do, and they often enter the ER lumen or membrane as they're being made. They also undergo initial modifications here like folding, adding sugar chains, or forming disulfide bonds.
And from the ER, where do these proteins go? To the Golgi,
Typically, yes, to the Golgi complex or Golgi apparatus.
which you described as like the cell's postoffice.
It's a perfect analogy. The Golgi is the stack of flattened membrane-bound sacs called cisternae, usually curved. It has distinct entry, cis, processing medial, and exit trans networks.
So it receives proteins from the RER.
Right, and then it modifies them further, maybe adding more complex sugar chains, phosphorylation, sulfation, and then it meticulously sorts these proteins and packages them into vesicles to be sent to their final destinations.
Like lysosomes or secretion or back to the plasma membrane.
Exactly, it's the central sorting hub. And interestingly, that cholera toxin we talked about, it gets processed and activated as it travels backwards through the Golgi and ER, a journey helped by a protein called ARF.
Wow, it all connects. Okay, finally, let's talk about the cell's internal framework, its backbone, the cytoskeleton. You said it's dynamic, not static.
Incredibly dynamic. It's not just scaffolding, it's constantly remodeling. It defines cell shape, anchors organelles, enables movement. It's made of 3 main types of protein filament.
Okay, what are they?
First, microtubules. These are hollow, cylindrical tubes made of subunits of the protein tubulin.
What do they do?
They act like railroad tracks inside the cell. Organelles get positioned along them, vesicles move along them using motor proteins like kinesins and dyneins that burn ATP to walk.
Like transport trucks on highways.
Exactly. Microtubules are also crucial for forming the spindle apparatus during cell division, pulling the chromosomes apart.
And this brings us back to Lotta T and colchicine for gout. It messes with microtubules.
Precisely. Colchicine binds directly to tubulin subunits and prevents them from polymerizing into microtubules, essentially causes the tracks to fall apart.
So how does that help gout?
In gout, microtubules are needed for neutrophils to move towards the crystals and, importantly, for releasing the inflammatory mediators that cause pain. Disrupting microtubules dampens that whole process, but it has a narrow therapeutic index. Microtubules are vital everywhere, especially in dividing cells, so the dose has to be carefully managed. Neutrophils happen to concentrate it a bit more, which helps target the effect somewhat.
Okay, so microtubules are one type. What else?
Second component, actin filaments. Also called microfilaments or thin filaments. These are thinner, more flexible helical polymers made of G-actin subunits.
What are they important for?
Crucial for cell shape, especially at the cell surface. They drive cell movements like crawling or migration, forming pseudopodia to engulf particles. And of course, they're essential for muscle contraction, working with myosin. They form that cortical meshwork just under the plasma membrane, too.
Giving the edge of the cell structure.
Right. And finally, the third type, intermediate filaments, IFs.
Intermediate. In size.
In diameter, yes. Between actin filaments and microtubules. These are different. They're more like ropes, tough, stable, fibrous protein polymers. Their main job is providing mechanical strength and resilience.
Less dynamic, more structural.
Generally, yes. Think of them as the cell's internal reinforcement cables. Examples include nuclear lamins lining the inside of the nuclear envelope, cytokeratins in epithelial cells making skin tough, and neurofilaments in neurons providing axonal structure.
What an incredible, intricate journey that was, through the whole cell from the outside in. It's just, well, mind-boggling how all these bits and pieces work together.
Isn't it? So coordinated, so dynamic, all performing life's complex functions, keeping us healthy.
Or, when things go wrong, leading to disease.
Exactly. And understanding the cellular world is absolutely foundational for medicine. The key takeaways really are these universal yet adaptable features of cells, the critical dynamic role of membranes and transport, and the specialized interconnected jobs of the organelles and cytoskeleton.
Seeing how they function normally is the first step to understanding disease.
Absolutely. You have to know the baseline to spot the deviation.
It really makes you appreciate the complexity. And as we wrap up, it leaves you thinking, doesn't it? Given this astonishing balance inside just one cell, what's the next big breakthrough going to be in understanding how these tiny malfunctions cascade into widespread disease?
That's the multi-billion-dollar question, isn't it? The more we learn about these tiny universes, the closer we get.
Well, thank you for joining us on this deep dive. This has been a warm thank you from the Last Minute Lecture team.
We hope you feel a little more well-informed, maybe even amazed, about the incredible world inside your cells.